Okay, so aside from oxygen binding to iron and other competitive inhibitors, like cyanide, okay, and carbon monoxide, there are also allosteric molecules that can bind to hemoglobin. Allo here, okay, allo here means other, and when you have allosteric ligand, okay, it's something that bind no to an allosteric site. If you're thinking of an enzyme, it's the non-active site. However, hemoglobin is a protein and not an enzyme, no, but it still has an allosteric site, which are not specifically on the heme group. Now, these ligands are involved in cooperative effects when oxygen binds to or released from hemoglobin. These allosteric ligands, okay, include carbon dioxide, which is released by the cells from respiration, okay, protons when you have low pH, okay, later we'll discuss further the effect of protons, and as well as 2,3 BPG. By the way, you can refer to these allosteric ligands as your allosteric effectors. No, again these structural changes, okay, can affect the hemoglobin's affinity to oxygen. Okay, so now let's discuss the effect of protons, okay, with that of hemoglobin. Okay, so dito na papasok yung Bohr effect. No? So in Bohr effect, okay, an increase in the concentration of the protons, thereby lowering pH, reduces the affinity of oxygen with hemoglobin. Okay, saan ba nanggagaling yung increase in proton concentration? Okay, so it came from the actively metabolizing tissue, thus acidifying its local environment. Remember here your erythrocytes contain hemoglobin and or hemoglobins kay and travel sila sa katawan. Now when hemoglobin reaches this acidic region, it releases its carried oxygen here because hemoglobin has a lower affinity to oxygen under these regions. Remember, okay, protons are allosteric effector to hemoglobin. Okay, and it attaches to a site other than the heme group. Now, those protons will protonate key amino acids, which changes the three-dimensional structure of hemoglobin, no, causing it to have a lower affinity with oxygen. Now, that is what is being shown in the figure. Now, so focus your attention here. You can see that hemoglobin or HB with oxygen molecules, okay, released it to an actively metabolizing tissue. In return, it picks up H plus molecules. Okay, now these H plus molecules came from the produced carbon dioxide, okay, during cellular respiration, or generated, okay, by the citric acid cycle. So remember here that the equilibrium, okay, between carbon dioxide and water to form carbonic acid, no? So your carbonic anhydrase here, so your carbonic anhydrase here facilitates the rapid interconversion of carbon dioxide and water to carbonic acid. Let's apply here our knowledge from before. Diba? So the pKa of carbonic acid is 6.35 and the physiological pH is around 7.4. So pH is greater than pKa. As a consequence, the dissolved carbon dioxide will exist in its deprotonated form. Now, so, or your carbonic acid rather, K, will exist in its deprotonated form. So you have your HCO3 negative or your bicarbonate form or bicarbonate ion. Now, releasing protons. So doon nang galing yung protons or yes, yung protons. Okay, so note here that during the conversion of carbon dioxide into bicarbon or pwede rin natin sabihin ganon, 'di ba? Your carbon dioxide into bicarbonate, K, or carbonic acid into bicarbonate, K, the liberated hydrogen ions here are buffered by hemoglobin, no? So this buffering helps maintain the normal pH, no? So this is reversed in the pulmonary capillaries. Okay, so going back to hemoglobin, no, which carries protons, okay, it will go back to the lungs, where there is already bicarbonate from the blood. Now, the presence of oxygen tension in the lungs would force your heme group to bind with oxygen, and as a result, okay, the protons now will be released to the lungs. Now, upon release, it will react with the bicarbonate ions, okay, to form carbonic acid. Now, the same enzyme, carbonic anhydrase, will facilitate the rapid interconversion of carbonic acid to water and carbon dioxide. Since we're already in the lungs, carbon dioxide will be exhaled. So there, that's your Bohr effect. By the way, this is only applicable to hemoglobin, no? Although myoglobin and hemoglobin are quite similar. Okay, so some trivia here. Hemoglobin can also transport some carbon dioxide directly. However, when its concentration is high, it combines with some free alpha amino group to form carbamate, which binds to the alpha amino terminal end of each globin chain, which in turn no, forms a carbamino hemoglobin. And there, kasama na siya papuntang lungs, no, where it will be released, uh, uh, where it will be exhaled. So, carbon dioxide is carried as carbamate in blood. Okay, so let's just have a run through of Bohr effect here. Okay, so from metabolizing tissue, okay, you have an acidic environment. So oxygen will be released here and the protons will be carried to the lungs. So parang exchange sila. Since there's plenty of oxygen molecules in the lungs, they will be carried and the protons will be released. Okay, now your protons will react with bicarbonate ions from blood, no, to form carbonic acid, and then carbonic anhydrase will catalyze the formation of water, okay, and carbon dioxide. Okay, and where the latter will be exhaled. Now here's the now here that's na nasa lungs na sila, dun talaga dapat i-release or dun talaga nare-release ang hydronium ions or yung H plus mo para ma-exhale because this is the organ that has high concentration of O2. So hindi siya basta-bastang marirelease kung saan kasi nga dito dapat mag-react or ma-convert yung H plus mo to pabalik to carbon dioxide. Okay, this slide shows you another ligand that binds with hemoglobin. If you remember, this is one of the allosteric effectors of hemoglobin. No? So your 2,3 bisphosphoglycerate. Now again, I just want to reiterate that binding of an allosteric effector, okay, to hemoglobin causes structural change. And that consequently, yung change na yon would consequently affect the oxygen binding capacity of hemoglobin. Okay, your BPG, okay, binds with hemoglobin via electrostatic attraction, specifically between the positive charges on the proteins and the negative charges on BPG. I hope nagkakaintindihan pa rin tayo na BPG is charged because it's in biological setting. No? Di ba sinabi na natin yan before? Okay, what is it for ba yung BPG? Okay, so BPG here stabilizes the T state of hemoglobin and promotes release of oxygen. Put it this way. Remember, if hemoglobin is in T state, it means that there is little to no oxygen. And if that's the case, okay, hemoglobin will not be inclined in releasing its carried oxygen. Now, BPG here lowers the affinity of oxygen with hemoglobin, no? So making it easier for it to release yung kanyang daladalang oxygen. So this is particularly useful for those living at high or in high altitudes since we know that thin air yung present sa higher altitude, no? So you have here considerably less oxygen. So it follows that people living in highlands have less oxygen intakes, and thus their hemoglobin is always in T state. Okay. However, why is it that they can still continue in their everyday life? It's because they have adopted to that environment, no? So their body ramped up the production of BPG so that they have high concentration, no, or high concentration of BPG as compared with those living in lowlands, no, and that's what enabled them in living in that kind of environment. No? So this is the process of acclimatization. So ito yung ginagawa ng mga mountaineers, 'di ba? 'Di ba nagse-set up, they set up different stations at different altitudes and they stay there for a while. For their body, yung reason nun is that for their body to adjust and increase the concentration of BPG so that oxygen can be easily released to the tissues that need it, no? So, parang pinapag-move on niya yung T-state ng hemoglobin mo para madaling pakawalan yung oxygen na dala-dala niya, no, yung mga saloobin niya sa life. And then, some trivia here, okay? Smokers also have elevated BPG because the oxygen carrying capacity of their blood is reduced due to the inhalation of carbon monoxide in tobacco smoke. Okay, so the non-covalent interactions present in proteins are very weak, and as a result, it's not surprising that the structure of proteins can be disrupted. Okay, so the disruption or the unraveling of a protein is called denaturation. Okay, here the protein loses its structural order. No, it loses its quaternary, tertiary, or secondary order, or any combination of these. Okay, which results in the loss of the biological activity of the protein. Because again, we know that these structures are what gives a protein its biological activity or function, specifically yung tertiary natin. Okay, and kakadiscuss lang natin in the case of hemoglobin na talagang may epekto yung change sa structure niya. So, denaturation can be brought about by your denaturating agents. So, these include heat, okay? What happens here is increase in temperature causes vibrations, okay, within the molecules. And the energy coming from these vibrations can become too great, okay, or too much to handle that it can disrupt, para sa protein ha, that it can disrupt the tertiary structure. Okay naman, next we have extreme pH, extremes of pH. Okay, meaning either high or low pH, no? So changes in pH, okay, affects the charges on the side chains, no, and so the electrostatic interactions that would normally stabilize the native conformation of the protein are dramatically reduced, okay, thereby leading to denaturation. As you go along, we'll learn that there are specific pH or optimal pH that proteins or enzymes, okay, function well. We also have here detergents. For example, your SDS, sodium dodecyl sulfate. Now, so detergents, as we have discussed before from micelles, okay, from micelles, kung naaalala pa natin yun, no, yung SDS from micelles. Now, those micelles tend to disrupt the hydrophobic interactions within the protein. Note here that when a detergent is charged, it can also disrupt the electrostatic interactions. Okay, now move naman tayo sa urea and or guanidine. Okay, so these two are also included here, no? So both of these compete with the hydrogen bonding formation for amino acids, no? So they can also disrupt hydrophobic interactions in the same way as detergents. No? Same ng ginagawa ng detergents. And lastly, we have mercaptoethanol. So this agent reduces the disulfide bonds in protein or in a protein. No? So there. Okay, so this is just a visual representation of the denaturation of a protein. Okay, so if you will analyze the figure, you can see that indeed the native conformation of a protein is disrupted. But if you will look at the denatured protein, okay, you can see that the primary structure or the amino acid sequence is still present. Okay, we can therefore say that a denaturating agent does not affect the primary structure. Now, what is the implication of this? If you will recall, okay, we said that the primary structure of a protein conveys all the information okay, necessary to produce the correct three-dimensional structure. Ibig sabihin, okay, the native conformation of a denatured protein can be recovered when the denaturating agent or the denaturating conditions are removed. So, hindi ibig sabihin, okay, na kapag na-denature yung protein, wala na siyang pakinabang, no? So, you can still recover its complete three-dimensional structure. You only need to remove the cause of the denaturation. Okay, so the process whereby the denatured protein reverts back to its native conformation, okay, under certain conditions, okay, is referred to as renaturation. Okay, so shown in the figure is the denaturation by adding urea and beta mercaptoethanol to ribonuclease, and renaturation of the same by removing the denaturating agents. Okay, so protein folding dynamics is quite a lengthy discussion. So I'll just include here infos that are, I think, important. So to accomplish correct protein folding, you need to have the correct primary structure, as what we have been saying repeatedly. Okay, so the driving force, okay, in protein folding into its specific three-dimensional structures are hydrophobic interactions. Now, so recent studies have found out that proteins tend to be folded in such a way that the non-polar hydrophobic side chains, okay, are in the interior of the protein, while the polar hydrophilic side chains are on the exterior of the proteins, no, and are accessible to the aqueous environment. So, what makes the hydrophobic interactions favorable? No, it has to do with spontaneity. Okay, so the entropy of the universe, okay, increases when hydrophobic interactions occur. So you can see in the figure that folded proteins have lower energy as compared with it, no, being unfolded. Note that the bumps here, okay, so these yung mga yan, okay, represents energy barriers, and the folding that will take the fat, okay, so rather, and the folding, no, will take the path of the least resistance to achieve its final native conformation. Okay, so to ensure correct protein folding, no, so special proteins called chaperones help in this process such that correct and timely folding of proteins are achieved. Now, how does it work ba? As the name suggests, okay, it's like a protector of protein. No? So it prevents a protein from associating with other proteins, okay, na hindi naman niya dapat samahan, or keeps it from associating with itself in the wrong manner. Okay, so we also have here chaperonins, no? It's a class of chaperone that assist in protein folding as well, no? So they provide proteins with favorable conditions for correct folding. So pag chaperonin, meron kang parang lalagyan dito, then papasok din yung protein, then mag-close siya para mag-fold din yung protein correctly, and then para walang influence ng outside world. So, after folding, so yun na, open na ulit siya, and then meron ka ng correctly folded protein. Okay, so we have been talking about protein folding for a while now. So, ano nga ba yung importansya nito? Kasi, failure of your protein to fold properly produces inactive and toxic proteins na that malfunction and cause a number of diseases. In addition, okay, correctly folded proteins are usually soluble in the aqueous cell environment, or they are correctly attached to the membranes, no? So note that wrong folding may cause interaction with other proteins, therefore producing aggregates, which is very dangerous because accumulation of these aggregates, okay, lead to several neurodegenerative disorders, no? So which we will be discussing on this slide. Okay, so protein misfolding causes the following diseases, among others. Okay, first we have prion disease. If you're familiar with the mad cow disease or the bovine spongiform encephalopathy, okay, and its human counterpart, Kuro disease, or human spongiform encephalopathy, okay, so because the causative agent, okay, for these diseases are proteins called prions. Okay, so prion proteins or PRP, okay, are actually natural glycoproteins, no, that help guide cell that help what do you call this, guide cell maturation. However, okay, when normal prion folds into an incorrect form called PRP raised to SCK, problem occurs because misfolded prions have the ability to trigger or infect, so to speak, other normal proteins into assuming a misfolded shape. So it's like a bad role model na pag napatabi sa isang normal protein, they can convert it into abnormal forms. So, di ba, nakakatakot. So, now this change can be propagated in the nervous tissue, which causes it to have holes, no, spongy form. Now, this is where the neurological problems take place. So dagdag ko lang dito, yung Sakuro disease, okay, Kuru is found among people from Papua New Guinea, okay, who practice a form of cannibalism. So sa kanila kasi, they eat the body of dead people as part of their funeral ritual. Now, they were shocked because women and children, okay, are getting sick, no, exhibiting symptoms like headache, okay, difficulty in walking, tremors, and muscle jerks. And coordination problems, no, that eventually became severe. Okay, now the reason for that pala was because only women are eating the brains of dead people. And we know that prion diseases are transmittable. Okay, so much for that. Now we also have Alzheimer's, Parkinson's, and Huntington's disease or diseases. Note here that unlike prion diseases, these are not transmissible, no? Pag kinain mo yung ganung bagay. The leading cause for these diseases involve plaque forming proteins. Okay, remember we mentioned that misfolded proteins form aggregates, and this in turn affect brain functioning. I'm sure you're familiar with Alzheimer's, okay? So this is associated with progressive memory loss. Okay, Parkinson's on the other hand is a disorder that affects movement, and Huntington's naman is a disorder that causes the progressive breakdown of nerve cells in the brain. Okay, so imagine these diseases, okay, among others, are caused by protein misfolding. So, di ba napaka-importante na correct yung pag-fold ng protein natin. So, okay, that ends our lecture for topic 2.2, no? So, kindly answer the exercises from your copy of this slide. So, I'll just upload the answers for those exercises on our Facebook page. Okay, so thank you, good luck, and see you on the next lecture.